Doctors Longo and Roberts discuss the current status of gene therapies in rare neuromuscular disorders in this 8-part podcast series. This is derived from the symposium that was presented at World Symposium 2025 in San Diego, California on February 4 through 7, 2025, and is intended for healthcare professionals only. This podcast includes information about investigational compounds that do not yet have a regulatory approval or authorization for a specific indication. The safety and efficacy of the agents under investigation have not been established, and contents of this podcast shall not be used in any manner to directly or indirectly promote or sell the product for unapproved uses. The views, thoughts, and opinions expressed in this presentation belong solely to the author and are subject to change without notice. The contents of this presentation do not constitute an endorsement of any product or indication by Astellas. In this part, Dr. Roberts will discuss immune responses and other safety concerns related to gene therapies.
Undoubtedly, the immune system is a major issue in these patients, and it would be fantastic if we could immunotolerize our patients and indeed prevent the rejection of the therapy. We've talked about the fact that these are viral vectors, and of course, there may be high seroprevalence of antibodies to these viral vectors, and it's very important in the pre-screening of patients who might be eligible to understand that at the beginning. And these, of course, can have developed over the years, and of course, can be part of immunological memory, and therefore extremely difficult and probably impractical to actually shift. On giving the treatment, though, as I think we're all aware, there is this problem of the innate immunity and potential, therefore, for acute toxicities, and then a learned or adaptive response with cytotoxic T cells and antibodies, which may, of course, become high-titer neutralizing antibodies, and potentially the antibodies not only against the viral vector, even the functional protein, even the transgene, are all theoretical possibilities with time. The capsid, the transgene, and even the protein product can all potentially induce an immunological event. And of course, all of these would lead to both potential patient changes and a lack of efficacy of the treatment. And indeed, there have been some serious and indeed fatal problems in the gene therapy development program, as I think we're all aware, though many of these are now thankfully been overcome. So spinal muscular atrophy has a gene therapy which is licensed, but there were early patients who actually had significant problems. So a patient of just 6 months of age who developed kidney failure, to other patients who actually developed liver failure. In Duchenne muscular dystrophy, a very common condition, again, there were significant issues, and crucially in these patients who all have cardiomyopathy, it was heart failure and cardiac arrest that were big concerns, and pulmonary edema. And this was seen even with a CRISPR-based technology. And as perhaps is best known, but has been addressed, the X-linked myotubular myopathy patients, 4 patients died, and crucially, quite a long time after the gene therapy, emphasizing the need to monitor these patients extremely carefully. And these patients died of cholestatic liver failure, albeit that they had a degree of liver dysfunction. And that's changed our screening, of course, of patients. We're now all looking in myotubular patients for liver involvement and Rett syndrome as well. Now, these immunoprophylactic treatment regimes to hopefully try and reduce the immunological reaction against the gene are certainly evolving. Pre-treatment with rituximab can certainly be a very useful treatment, and certainly if you compare, as this good study has, the use of steroids alone to those patients receiving agents such as steroids, but also rituximab and sirolimus, one sees a much lower incidence of these immunological reactions, accepting that these treatments in their own right have some risk to clinical patients. And this is just a summary of some of the other immunosuppressive regimes used in other disorders, for example, spinal muscular atrophy, but Pompe and MPS as examples of LSDs. And certainly these regimes will continue to evolve and are going to be very important in seeking to make sure that these treatments are effective. And it reminds me somewhat of what's happened with enzyme replacement therapy, that the use of these immunological strategies in infants has revolutionized the utility of those treatments in early patients.
In the next part, Dr. Roberts will discuss lessons learned from gene therapy trials.